Know that genetic diseases can have devastating effects, but do we really know their foundations and how they happen? My name is Gerline, and this is the focus of my project. So before we get into genetic mutations, let's first start as how the genes and DNA are actually important to our bodies. So first off, we know cells, which are the building blocks of life. Cells, eukaryotic cells, have chromosomes in them, which have DNA, deoxyribonucleic acid. Deoxyribonucleic acid is supposed to be a code to provide instructions for our bodies and how parts of our bodies are supposed to function and be structured. So DNA will have 4 bases or nucleotides, which will then create the code, and the code will be transcribed into RNA, and from RNA it will be translated into an amino acid. So when it is translated, it will have 3 nucleotides that will create an amino acid codon. So multiple codons can code for the same amino acid, and 3 nucleotides make up 1 codon. So these amino acids will then form peptide bonds, and then multiple peptide bonds will form polypeptide chains. And then with many polypeptide chains, you can have a protein that is supposed to do great things for your body. So the focus of my project is on type 1 neurofibromatosis, and this is a disease that is normally identified by its symptoms of neurofibromas, optic gliomas, Lesch nodules, and cafe au lait spots. So neurofibromas are basically tumors that grow on nerves in our body, and optic gliomas are basically tumors that grow in the optic pathway that connects our eyes to our brain, the visual cortex of our brain. And because of this, there are also symptoms of neurofibromatosis type 1 that can be or along the lines of attention deficits and learning disorders. Lesch nodules and cafe au lait spots are just irregular growth of cells and tissues. So let's talk about the Nf1 gene that is mutated to cause the disease. So the Nf1 gene is spread over 280 kb of genomic data, and because it is such a large disease and has gene and has such a wide spectrum of mutations, it is very hard to detect mutations in the first place, and let alone detect mutational hotspots or amino acids that are commonly mutated. Basically, the Nf1 gene is supposed to provide instructions to make the protein neurofibromin, and neurofibromin is supposed to act as a tumor suppressor in accordance to the RAS protein. The RAS protein is supposed to be responsible for cells and tissue growth, and neurofibromin is supposed to help prevent this protein from growing cells or tissues rapidly or irregularly. And because in the disease, this protein is now shut down and dysfunctional, there are times where the cells or tissues will grow rapidly or irregularly, which is where neurofibromas, optic gliomas, Lesch nodules, and cafe au lait spots come from. The reason they are growing on nerves is because neurofibromin is often found in oligodendrocytes or Schwann cells, and basically other neural tissues, which is why it often occurs on nerves or in optic pathways, etc. Neurofibromatosis type 1 occurs in 1 in 3,000 to 4,000 live births worldwide, and it can be a very devastating genetic disease because the tumors in it are not always benign. They can be malignant as well, and sometimes optic gliomas that are featured in this disease can also lead to partial, peripheral, or central, or even just complete blindness. And so I had done my report and procedure, and I had focused it on the 10 pathogenic alleles that I had chosen, and then figured out a procedure for. Basically, at first, I decided how the disease was actually caused because of the mutations, and then I had gone through or delved in deeper into how the DNA that was mutated transcribed and translated into basically the wrong amino acids, which then led to the protein being dysfunctional. And basically, the procedure that I had followed for the analysis was to figure out how the mutated amino acids were going to further affect and then I had figured out how the protein structure was greatly affected, including its function, by the mutated amino acids being different and the types of mutation that would occur in order for it to be most severely affected. And I had also delved in deeper into how the mutated amino acids were different from the originally intended amino acids when it came to how the protein was supposed to function. So The databases I used for this project were the National Library of Medicine and also the National Center for Biotechnology Information. I had picked out 10 pathogenic alleles for the neurofibromatosis type 1, and I had found out how the amino acids in it were different. I had also figured out that during my analysis that the structure of protein may be very severely impacted because different amino acids may only interact with other amino acids because of either charges, polarities, etc. There are also times that are mutations such as nonsense or missense mutations where nonsense mutations will often lead to an amino acid codon instead coding for a stop codon, therefore chopping off a large part of the amino acid sequence and also removing a large part of the protein, therefore leaving it dysfunctional and also very thrown off in structure. There are also mutations such as conservative mutations and non-conservative mutations. The amino acids will be different, but will be of the same type, so they don't really affect the function or the structure of the protein. There are also frame shift mutations where a nucleotide will be added and therefore will shift the entire frame of the amino acid sequence and will lead to entirely different amino acids being made for the protein, and that will obviously affect the protein in very bad ways. The way an amino acid change may affect a protein function will be for example, non-conservative mutations are supposed to be where amino acids will change, and they will change or be mutated into an amino acid that is completely different or even the exact opposite of the amino acid that was originally intended. So for example, in diagrams over here, there are charges where if, for example, the charge turns out to be the opposite of what was originally intended, it will interact completely differently to its environment. There are, for example, hydrophilic and hydrophobic amino acids that will completely interact completely different to its environment if they were mutated to be the exact opposite of its properties. There are also these structures of proteins where, for example, if a protein or amino acid were hydrophobic, it will decide to stay within hydrophobic cores of a protein instead of on the surface, where, for example, it was originally supposed to be intended to stay on, and that will obviously throw off a little bit of the structure of the protein. So the basic purpose of the entire project was that to figure out how even the slightest changes would affect an entire protein from being made, and also even create an entire genetic disease, which we had decided to eventually manage to figure out in our project.